To continue with paper 2 practice, what we've left off on then is having to describe or create a typical lab experiment or scenario where you're going to test out maybe like a limiting factor on photosynthesis. You guys covered limiting factors in the 3.2 notes, and those were like right before and after spring break. Things that can be limiting factors for photosynthesizers is going to be the amount of light intensity, the temperature, carbon dioxide, but because we are always like combusting fossil fuels, it's really not going to be a limiting factor, and nutrient availability, right? So too much obviously can cause an algal bloom. It could cause like cyanobacteria to increase in toxicity, or increase in population, and they will end up synthesizing toxins, can, get in your trophic levels and cause other organisms to suffer because of that. Anyway, so this one is going to be investigating the rate of photosynthesis. And so to do that, we're going to obviously need light. You may be asked, maybe not to do this same one, but maybe investigate the effect of temperature on the rate of photosynthesis, or maybe how salinity affects something. And if they're asking if how light intensity affects photosynthesis, then light is going to be what you are manipulating, or what you're changing, what you're changing the amount of. Similar to this one here, temperature and density. The temperature was what was changed, and then what was recorded, or the response, was the density. In this situation, you're going to change the distance or the amount of light. You wouldn't want to go and make it as far as to say like different types of light bulbs. The easiest thing is to change the distance of it. Something I think is very helpful, like I showed on the sampling practice that I made for y'all, is like this one right here. When they're describing a situation, especially in a testing scenario, we all automatically kind of feel like, oh my God, like a little panicked. Draw it out. What do I say? When in doubt, draw it out. You can still get marks for, depending, if they're allowing it or not, but you can still get some marks for having a diagram or an annotated diagram. And I know for me, it's easier to see these things. So describe an experiment that you would use to safely test the following hypothesis. Okay, so things that we're going to want to make sure that we mention. Okay, I'll put it off on the side. For any of these, you're going to want a list to state what your, for dry mix, talked about it in the last video, and I talked about it at the beginning of the year. Your dependent, or the responding variable, the response is your answer, so what you would be measuring. It's going to be in the y-axis, and then there's your y. The manipulated, or independent variable, and if you're manipulating something, means you're changing it, is on the x-axis. And in this situation, we're going to be changing the light intensity. So you're going to want to state what the independent variable is. So we're going to say, like, we're going to need to describe like how we're going to do like different types of light intensity. So the independent variable, how we're going to measure it. So one of the things you're going to get through photosynthesis is oxygen. And so in this situation, we're going to measure oxygen bubbles. Okay. Within the independent variable, well, there'll be other little details we mentioned too. Okay, we're going to need to say like a method to record that. So if you were trying to see like how something grows or its height changing, the method would be measuring its height and maybe using like a meter stick. Okay, I'm getting on. So any two, it says on your mark scheme, and this is a new skill that's being tested. The mark scheme is saying state two control variables. And control variables, just to remind you, are things that you're going to keep the same, things that you're going to maintain throughout the experiment, to make sure that they are not a factor in your experimental results. So you would want to, like, for example, LED lights are really efficient because they produce more light than other bulbs. And let me rephrase that. They don't produce as much heat as other bulbs. You don't want to have maybe a light bulb that is giving off a lot of thermal energy, because then the temperature can change. If the temperature is changing, then you don't know if your results are because there's a change in temperature or because there's a change in light amount. Okay. All right. My afternoon classes were more full than this morning. I think there was another exam. And we did talk about this in the afternoon classes, like what kind of things would you make sure are the same in water? Okay. We're going to want to, of course, you don't want to just do something once and then take that for verbatim. We're going to want to mention something about repeats. Not unlike this one here, where we talked out using the quadrat. So repeating, counting, taking an average. So the repeats, once you have the repeats, you're going to need to take an average or a mean. Okay. You will get an extra mark, or this is, don't be intimidated by the lines. It's 5. I think you guys may have been intimidated by the lines on the question that said ways that nutrients can be put back into the ocean, but it's only 5. So you will get credits for other little details, like in this example, something that you did want to control was heat. So if you did mention, and I don't know like what this actually is, but so there's some screens that can limit the actual heat from transferring to your water to your variables, your specimen, and so it's just light. But for this one, we can just say like an LED light. So making sure that temperature isn't changing, but you have a lot of different options to get these kind of questions correct. So you're not limited to just 5 things. Okay, maybe a little detail about CO2 I can mention. And then of course, I'm going to refer back to this one that I lectured out already. New in paper 2, and I handed this out in class to you guys Thursday before Easter. For sampling techniques, one, you're going to need to talk about ethical practices. Also, safety measures. So in this one, you're using like a lamp. So maybe like don't burn yourself with a lamp. You're going to be using, in this situation, lamp, and it's going to be near water. So maybe let's not put the outlet near the water, or like any kind of like electrical safety. All right. Okay, so I did include a blank page here so we can kind of see what this is going to look like. In class, I know I said like maybe use like a tank, and then, but honestly, that's going to be too varied. I think there's going to be too many organisms in there, and ultimately that's going to be too big of a space for just this little thing. So a recommended way is to actually just use it in a test tube. So let me just draw out what this might look like real quick. Test tube, and we're gonna fill it with water. I don't imagine you have to talk about like putting it like in a clamp or like a stand so that it doesn't fall. I mean, you could say you're gonna put it in like a test tube rack, like literally like those like wooden racks. And you like put the test tube in it and it holds it. I don't think that's gonna be what's gonna get you all the marks though, like those details. Okay, and inside of it, we're gonna put like a piece of like a common one is called elodia. It's like pondweed. However, they say seagrass. Seagrass. Okay. So, yes. All right. So you could say like you could take like a strand of seagrasses and they have like a root structure. I'm actually going to draw that differently. It's what I showed you in class. We have seagrasses and just do a little bit correct. Remember, they have the long rhizome that connects them. Roots come off of it. And that helps them like not get pulled from their substrate. Okay, I'm gonna fill the water up a little bit more. And don't like if you miss like some details, that's okay. Like there are so many answer options for this. It's all right. Even if you say like just mentioning repeats, like you should be able to get some marks. Okay, so we have this in there, and then we're gonna put in our light source. I'm like just trying to imagine like what a here, I'll give it like a circular stand. Cool. Okay, and then in that, right, where I said like an LED bulb. All right. And now you will actually be able to see bubbles produced from this. And because they're a gas, they will rise. And that is not an accurate representation of how many bubbles that will come out, depending on the distance. We're gonna want to be able to recognize the distance, right? We could use a ruler, but we're not gonna measure in feet because we're not answering for America. Not only that, in science classes, you don't use feet. Okay. 30 centimeters are on one ruler. You won't ever have to remember that because you're going to have a ruler in front of you. Okie doke. And you want to see the effective light intensity. So perhaps you'll do this multiple times. So perhaps you want to, and you can like take this entire thing and just duplicate it. Copy. Okay, it doesn't have the bottom. I'm fine with it. Same thing. Sorry. Okay, so this is, we're gonna say it's about like, I know there's like a little gap here, but we'll say it's about 30 centimeters. That's not the same size, but 30 centimeters away. And here, we'll say that this, we'll do one like 30 centimeters, and then we can imagine that this one is going to be a little bit closer. We'll say this one's at 20. And then we'll do it all over again, and we'll put one closer. Just bear with me just so you can see it. Okay, this one's a 10, right? In real life, it wouldn't be so close like this, but I think you get it. So 30, 20, 10. Perhaps we also want to do like two more, right? So imagine there's also one at like 40 centimeters and then another one at 50 centimeters. Okay. And so you're going to let this like get acclimated to like the light, and it doesn't take long. It's just going to take like some minutes, literally, maybe like 5. And then it should be starting to produce bubbles, depending on its proximity to its light source. So we can already assume, just from common knowledge in photosynthesis, and it was already in your notes, that the closer you are to the light source, the more bubbles you'll produce in a certain amount of time, right? So the faster the rate of photosynthesis. And that is one way that we can be able to identify this. And so you'll be able to count the bubbles. You don't want to say like, I'm going to do this for an hour, because, well, now like you're just opening yourself up to a lot of human error. Like, it's, that timeline is not realistic. Same with, we needed to have a realistic distance on this. So you wouldn't want to say like, I'm going to do it from 20 feet. Like, are you? Like, I'm pretty sure that light's going to dissipate. So this is done better if it's like in a dark room, so it's not having any access to other ambient light. But as long as that's standard, the ambient light is standard, this additional light should also be able to show like a relative increase. Okay, so it's going to look like, and then you could say like, you want to see like the rate of photosynthesis, right? So rate is a speed, the amount something is done in a certain amount of time. So there will be a time factor. You want to see like if the proximity is going to cause it to produce more bubbles. So we will also want to mention that we need like a stopwatch, or like a timer. And then the idea of counting oxygen bubbles produced. All right. Last thing, just for imagining this, you don't want to just do this one time and say like, well, I did this, experimental setup once and that's that. Guess it works. So, doing this at the 30 centimeter mark, the 20, the 10, and then, 40 and 50 as well. It's not going to be legit. So you want to definitely offer some more, what do you call it? More trials. Put that there because we're going to want to mention that. Yeah, we'll want to have trials. So an idea of like repeating this. So maybe this entire situation that you're doing, you and maybe like another person, you know, you're doing it at the same time, so maybe in the same area, same place. You'll want to make sure that you get your seagrass from the same area. Those are like the control variables you want to try and maintain. You're going to do this whole setup maybe, let's say, 3 to 5 times. So like for 1 minute, you're counting bubbles that are coming out of this test tube, right, at the 30 centimeter mark. And you're going to do that one specifically, like bubbles at 30 centimeters produced in 1 minute. You can do that 5 times. So you'll have 5 trials. So maybe it'll look like when you record it. I got so lucky with that. When you record it, looks something like that. And then, you know, you can take this number here, this number here, this number here, this number here, number here, and you can add them up, divide by 5, and then you're going to get an average. And so now on average, you know that at 30 centimeters in 1 minute time, you're going to get, you know, this amount of bubbles produced. You will do the same thing. You don't have to draw this out by any means or make additional data tables other than the one that is just requested of you. Why? Curses. This one will be at 20. Okay, and then I'm going to add these 5 up, divide by 5, even average, for the bubbles produced at 20 centimeters. The averages are what you're going to use. You do the same thing right here at 10 centimeters. You're going to like have this set up. You can do it 5 separate times, or you can have this set up like at once 5 times. But then you would need 5 people to be counting the bubbles. You would need a lot of lamps. So it probably would be easier just to, I mean, it's only a minute, right? So you do it 5 times for your 30 centimeter mark, 5 minutes. 5 times for your 20 centimeter mark, another 5 minutes. 5 times for your 10, another 5 minutes. So right there, you're 15 minutes. And then the 40 and the 50, 25 minutes. Okay. So that's kind of like how I imagine it. And all right, we'll write it out. So let's first talk about like getting the seagrass. We'll want to use words like carefully, just some fun buzzwords. And I'll go back and highlight things that are like buzzwordy. There's also examples of this setup, I believe on like YouTube and other things like that. Okay, so it's carefully removed, or you could say the seagrass is kept submerged. So this mark is saying like how you're gonna either get it or how you're gonna like maintain it. Okay. Now we need to say the independent variable. And independent, again, is what you're manipulating. If somebody manipulates you, they're trying to like get you to do something like in like change, trying to get you to like to do something. They're trying to change something. If someone's a manipulative person, they try to get their way by manipulating you to doing something you normally wouldn't do. So this like changing. The manipulated variable is the thing that you're testing, what you're trying to test out, what you're trying to change to see the effect of. And then the response is how you're measuring. In this case, it's going to be counting bubbles. Okay, so okay. We can then discuss within that the independent variable, the light source, give it like an actual example of like maybe what those distances are. I mentioned doing this for population sampling, where I said like the quadrat size, or actually saying like you're going to sample every certain amount of meters, like, but it has to be something that's realistic. Those are little details. If you don't remember it, that's okay. This is 5 marks and you're like 2 on your way. Forgot how to count. And this is when you're going to use pen on as well. So you could, if you did make that mistake, just cross it out, put a 4 by it. Okay, so I just took care of the independent variable. I said the word carefully. Okay, dependent variable. We can just call it oxygen bubbles. Bubbles will be counted, or bubbles produced will be counted at 1 minute's time within the test tube, and you actually can see it like moving up, like this happens.
Meet your next pillow. Why do we make our pillow in the shape of a cube? I'll tell you. The problem with pillows is that you're trying.
Okay, that's one.
If you look carefully, you should be able to see little bubbles.
That's another one.
Coming up.
He's
you can
counting the
count
bigger
those
ones.
bubbles
There's a big
over
one,
a
2.
period of a minute.
Here's
One.
another big one.
Two.
Another big one. So the smaller bubbles that, and you can do this like just in a standing test tube as well. The mark scheme describes this method here that I'm moving.
Photosynthesis is the process by which
And in that one, they are like actually submerged. Now it's right here in the top, but they're actually submerging it and in a funnel, and they put a test tube over the funnel. And then as the gas moves up that test tube, it kind of like pushes the water level down. So that's where like they'll measure the actual like amount of gas produced, because the gas will pile up at the top, and effectively that pressure is going to push the water in the test tube further down back into the beaker, and so you can see the amount of gas that collects.
Two.
The bubbles that you see on the organism itself, or those tiny tiny ones coming up, are actually just like, from what I understand, what I believe, like it's going to be just like residual from it being like exposed to the atmosphere. It is not the oxygen produced. That's yeah, that's that. It has that waxy layer.
Three.
Another one.
Four.
You don't have to know like a typical result. So just so you can see that you can actually see them. Okay. Let's see. We're gonna say, all right, so we talked about the different distances. We'll mention each. Will be repeated. Ooh, it's nasty R. I'm just gonna say 5 times. Be repeated 5 times for each distance. Okay, so I said the repeats. I still need to talk about the controls. What we're not going to change, or what we're going to make sure is not changing. Okay, and then once we have, right, we did this repeat, right, we repeated it for each distance, and we're going to take the average. I'll mention the LED light. Ideally, I maybe could have put it like at that second mark, but I can still add it. It's no problem. I am at 6 marks right now. This is a total of 5. You'll need at least a minimum of 2 controls. Okay. So I said that counting bubbles produced. if I don't know that it's super necessary, but at that 1 minute mark for um, the the fourth mark, so will be counted for 1 minute. I don't know if you wanted to mention like stopwatch or a timer. If you were like trying to measure the effective temperature on something, you would want to mention like maybe using the like a thermometer, perhaps putting it on a hot plate, putting one on a hot plate or putting another one in like a cold water bath. Not that there's ice in it, it's like sitting in a bath of ice. Like the actual, maybe the beaker is sitting in like a larger bath of ice, right? And that'll like keep um, the entire beaker cold. Okay. All right, almost done. So things we want to keep controlled. We only want to change, so if we can imagine what's happening here, we only want to change the distance. So let's just consider like the water. So within the water, you got temperature. You want to make sure the temperature doesn't change. And then we can also mention the LED bulb for that. So temperature won't change. We want to make sure that it's the same type of water. It's like ionized water versus like tap water, or like if you're, whatever source of water you're going to use needs to be the same. You could maybe even say like make sure the pH of water isn't changing, but honestly, I mean it shouldn't if it's from the same place, but you know. The seagrass from the same location. The amount of water too you want to maintain. I said a minimum of 2. Location of seagrass sample. Maybe amount. Okay, so I got my 2 controls. All right. Okay, remember not to use the up arrows or anything. You could use H2O. And okay, so something that like can be used is baking soda, which is called sodium bicarbonate. In these situations, sometimes that's used to give the seagrass like a good source of carbon dioxide right away. So, and it'll like pretty much allow them to start photosynthesizing because they'll have light and they'll have water because they're sitting in water. So they're just going to need some CO2. Sodium bicarbonate is baking soda. It's a great source of CO2. I don't remember the formula for bicarbonate, so I'm just going to write it. All right, and then I'll add in a safety, but you only need 5 marks. You have quite a lot now. Ensure or be careful about, take care with, something like that. Ensure, electrical cords of the lamp are not near water. All right, so that's that mark right there. No matter the question, even if I'm like, I've never done this experimental setup before, I try and imagine it. I will try and like draw it out, like what could this look like? Okay. All right, so in the beginning of the year, I did go over this and how to make a table. I'll put it right here. There you go. So data tables. Okay, so large enough so that all entries can fit comfortably and you're not like writing them outside of the line. Great. It's bigger is better than having it too small and having to squeeze in the words. Okay, and you don't have to include assumed like results because you know if you haven't done it then you wouldn't know. Okay, include in the thank you. Include no. Include in the table of results columns for raw data. Use descriptive column headings. So describe what it was. So you were doing distance of the lamp from seagrass or lamp distance from seagrass. And we measure the distance in centimeters. Any required units. You don't put units in the body of the table. So an example right here. So temperature of water measured in degrees Celsius. So distance of seagrass from lamp or lamp distance from seagrass in centimeters. And then the y-axis, the response, is going to be like the amount of bubbles produced in a minute. So we'll say amount of bubbles or number of bubbles produced / MIN for the minute. Or if you're a fan of doing that, you could put amount of bubbles produced minute to the -1 if you don't want to use the slash. Or you could also always write PER minute. And in these tables, like you never see like degrees Celsius, degrees Celsius, degrees Celsius. It doesn't happen because you already have it there. Okay, use a pencil, use a pencil. All right, use the space below to draw a results table that you could use to record your results. Include full headings in the results table, but do not write in any results and use a sharp pencil. Okay. I don't have a ruler, but I mean to do it on here, but per the mark scheme, you do not need to include like multiple, shoot, let me try this ruler. You don't have to include like 5 boxes, right? To like, if you were gonna actually pretend like you were gonna record data, it says ignore that, but you wouldn't know that because you don't see the mark scheme ahead of time. So I'm actually using a ruler, but this is gonna be bigger than necessary. It's not anything you would lose marks on because I really just want to be able to fit in my my table headings comfortably. What's this? Bum, ba, and across. Okay, I'm gonna put a line down the middle, or if you want to like put your heading in the side on the left first, and then yeah, put it in the side of the left first, just make sure it fits, and then you can put your line down. But okay, this is going to be distance of the land from the seagrass. Measured in centimeters. All right, and if I'm going to erase this, but if I were to continue it, right, I would have the 10, 20, 30, 40, 50, obviously with lines going across. We're here. In a minute. Or you could have also written like, but you know, it's not my forte. My fave. Great. That part's done. You don't have to by any means. But sometimes they may have like an additional table put in. Or like you could go next to it and then do, or even like right here, like an average. And so you would have like the amount of bubbles. So I'm just going to go 8, 6, 4, 2, 0 maybe. Right. And then you would do an average, 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 average. Right. No, I'm field stupid. Never mind. You wouldn't do that because you are including the averages. You would take the averages. You already did it because you did it right here for each one. You're going to do it 5 times. Ignore that. Okay, use the axes below to sketch a graph of the results that you would expect if the hypothesis was correct. Well, let's go back and look at that hypothesis again. Light intensity will increase the speed or the rate or the amount of photosynthesis in seagrass. That is what we should assume. If they do ask for a null hypothesis, I'll mention it again. I mentioned in the sampling video. A null hypothesis literally means the not hypothesis. And that essentially will read like there is no correlation between the rate of photosynthesis and light intensity, or light intensity has no effect on the rate of photosynthesis. I have to erase it all because I know it'll bug some of you crazy, and I know it'll bug me crazy. Okay. I'm gonna put like a 0 down here. Hold on. What are we doing down here? This was your X. Okay. So this is the distance, identical. Distance of lamp from seagrass. All right. And it says you're assuming that it's going to be correct. Okay, so we would say like, you would think light intensity increases, the rate of photo or gas bubbles produced increases. Or we would have 0. And again, you don't have to, it says include labels on both axes. You don't need to include a scale. But I'll go over here to 50. Okay, so at 10, where it's closest, we could assume down here is low, up here is going to be high, no matter what it is. That's just how a graph works. We would have a bubble here. And it says sketch a graph, so you wouldn't necessarily have to, when it says sketch, you don't have to use a ruler. You don't. So, yeah, but I'll show you what that line should end up looking like with some fake numbers. 20 should go down a bit, 30 even further, 40 even further, 50 even further. Okay, and so your line would look like this. And I didn't even touch those points. We'll pretend I did. But that's the sketch. The graph sketching means you don't have to use a ruler. Like I said, sketch a pyramid of energy, no ruler. Explain. So you got to say, so explain is like, what do you see and why? More so, why? Why is this happening? Why would these be the results? Explain the relationship you have shown in your sketched graph. Okay, so we're gonna, like I showed you before, if it was like describe the relationship, this one you need to say what the essentially what the relationship is, and then why is that happening? So give a reason for. In terms of your graph, let's not do this, like bring it all the way down. Because then you're saying there's 0 bubbles produced at that amount, which isn't going to be like legitimate. Okay, so an increase in light intensity R8, right, of photosynthesis, or as the light intensity increases, the rate of photosynthesis increases. Got to remember the why. Mark's scheme says because there's more energy available which kind of like gets me triggered because there's so many times in previous mark schemes they were like, oh, the student said energy, really? And they didn't specify what kind, no marks. So like thermal energy, light energy, chemical energy, like for food, or like when you do respiration and then you get out ATP, like chemical energy. And they were so picky about if you didn't say what type of energy it was, then you wouldn't get anything, which is very Cambridge of them. So even though it says due to more energy available, I don't know. Yes, okay. Okay. I feel like the fact that they said if the hypothesis was correct, the hypothesis was light intensity and then the rate of photosynthesis. So I feel like perhaps it's not based off your own. Use the axes below to sketch a graph. The results you would expect the hypothesis is correct. This one I did is like based off your own, but what you've seen in your notes before is like down here. So we would go rate of photo. That is the hypothesis, and that's pretty much what they're asking. And then here, oh gosh, no, that's actually what you're testing, or that's the what you're measuring. What you were measuring was the speed of photosynthesis, and what you were manipulating was light intensity. So you have this in your 3.2 notes, and then the line would go like this. And then it does level off. This one, the first one that I did is based off like the data you actually had. But I guess it says use the axes to sketch a graph of the results you would expect if the hypothesis is correct. Include labels on both axes. So yeah, I guess that's like a little trick there is to go back to the hypothesis and you're saying light intensity will increase the rate of photosynthesis, where light intensity is your X and then the rate of photosynthesis is what you were trying to measure, your Y. And then we just designed a way to identify the rate of photosynthesis. So this one I actually feel more confident about. And it does like level off here because at some point it's like, okay, well, I've increased the light so much and I keep on increasing it. You know, your chloroplasts are saturated. They're full of light. Like what do you want them to do? They can't take in any more light. They're at capacity. You don't have an unlimited amount of chloroplasts in that, like you don't have an unlimited amount of chlorophyll molecules. So something that could change in that, like that's where you could be like, maybe at this point I could maybe increase the temperature and speed up enzymes so that the chloroplasts will like, I guess like filter through or absorb and move through the light faster. The light can be used to actually split the water molecule, just like an A-level topic. Maybe there needs to be like an increase in nutrients. You wouldn't say you need to have more water because they're obviously in water. And if you gave them a good amount of CO2, then you wouldn't want to say they need to have more CO2. Just thinking out loud, just Ed Sheeran. Good. Well, I'm glad I caught that. So that's based on the hypothesis. And this is common. Like if you look this up anywhere like online or even in your 3.2 notes, this is like the generic go-to graph for that. I mean, but the one I did like is based on your hypothetical data. Okay, I'll do this one and then I'm gonna go pick up olives. So photosynthesis can be used in phytoplankton, phyto leaf-like plankton floaters can be used to investigate the effect of light intensity on the rate of photosynthesis. Phytoplankton are trapped, so they are in there, inside gel-like substances, creating small beads. So they're inside of there. You see, some of those gel beads will contain phytoplankton. Suggests 2 advantages of using the gel beads rather than seagrass to investigate the effect on light intensity. And it suggests 2. Having them in gel beads, one, like they're trapped, versus using the seagrass. Let's see. You don't have to go into like a natural habitat to get it, so easier to obtain or to get samples of the organism. With that, it's like the amount inside of it is easier to control. So amount of sample or organism is easier to control, like versus ensuring you have the same amount of seagrass leaves or macroalgae. They're called blades. Mm-hmm. And you know, like, well, once I'm imagining it's like would be ordered or yeah. So like you'll be able to if you wanted to have like the phytoplankton, they're easier to manage. They're easier to handle with them being inside that gel bead. Easier to manage. They don't require as much care at all. Their like environment or better like their surroundings are standardized, like they're all the same. Like the surrounding, what's inside of the gel bead that surrounds them is all the same. Okay, fun, reading a graph. We'll color code that. And then we got another graph about the hydrothermal vent. Oh, sorry, it's the abyssal plain. I know the other questions ask about hydrothermal vents. This is the hydrothermal vent. Curses. This is just the abyssal plain. Abyssal plains can happen at transform boundaries. What was the other one? Divergent? Like on the outside of a divergent boundary? Okay, great. Great, great, great. That's a big question. I'll do this one really fast. So if you had to change a variable, this is from 2014, paper 2. So single cell marine algae, Isochrysis was their species name, are grown on a large scale for aquaculture, for like mollusks and fish larvae, so things that are like grown in aquaculture, like fish farms. An experiment was carried out to investigate the effect of temperature on the growth of that algae species. In the experiment, samples or cultures of that algae species were grown in test tubes containing a culture solution of mineral salts. These solutions including magnesium and phosphorus. Okay, why do organisms need magnesium, especially in algae species? Chlorophyll. Phosphorus. Phosphorus is needed for ATP. The phospholipid bilayer, which is cell membrane. They have a cell wall and a cell membrane. DNA, RNA, proteins, or with that, amino acids, because amino acids make proteins. All of the test tubes were illuminated, so lit, with fluorescent lights and incubated, so kept warm, at a range of temperature from 14 to 34 degrees Celsius. And then give a time factor. So after they want to see like an effect. After 10 days, the cells were counted. Okay. Using a counting chamber and a microscope. Literally, this is like a microscope slide. And in the middle of it is microscopic like quadrats. And then imagine this is what you do random sampling with. So you're like, ahead of time, you would say like, okay, I'm going to count this one and 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 this one, right? And you count the amount that are in there so you're not counting all of them. Here are the results. Notice these are 10 to the 4, so the amount of cells per cubic centimeter. So something like that, the amount that's in that area. Why were the cultures illuminated during the experiment? So why did they require any sort of light? Organisms that do photosynthesis require light. So that would be why, so they can photosynthesize. Require. Why would it need magnesium and chlorophyll? So we're just gonna go with what I told you before. Any of these? That's not feeling right. Describe the effect of temperature on the growth of Isochrysis. All right, so look at your numbers. Describe the effect of temperature? You're going to say as the temperature, this is your X, this is your Y. We'll just start that sentence right now. And you don't have this blank copy, but it's just an example. As the temperature, whatever it does, the temperature is going from 14 to 18, 22. It's increasing. The number of cells watch this number. Okay, you have 250 times 10 to the fourth. And if you're thinking, yeah, well, that's what you should be looking at. But if you're thinking 250, then 120, you're like, why did it go down? It didn't. You're adding a zero. 250 times 10 to the fourth is really 250. And then you're going to add in 4 more zeros. 2.5 million. This one's going to be like 12 million. So we have an increase. And then we have 120 times to the fifth, 614, another increase. 614 to 845, another increase. 845 to 722, decrease. 722 to 230, decrease. So based on like your biology knowledge, and that is a prerequisite for this class, you can see that their threshold is going to be like where they're maxing out is 26 degrees Celsius. What's happening right here, the cells are decreasing a lot. That means that they are denaturing when enzymes get too hot, or they're, and we get, that's what happens with the fever for us, or if they're put in a pH that is not their ideal pH range, whether it be high or low, that will cause cells to denature, which essentially means the proteins that make up those enzymes. And I didn't mean to say cells, but the proteins that make that those enzymes are. Enzymes are proteins that speed up reactions. They unwind and they lose their shape. So now they're like not effective. So the number of cells, and we're just going to write exactly what we witnessed. So they start to increase, right? So it goes up and then it comes down. So we want to talk about this increase. And then what happened here? Number of cells increase until is it 30? Yep, 30 degrees Celsius. Come on. So as the temperature increases up to 26 degrees Celsius, the number of cells increase until 30 degrees Celsius when we see the cells start to decrease. We can also reference that in terms of enzymes, this is what we would call an optimum temperature. Ours is 98.6. Okay, and then if you wanted to say like the amount that it decreased, so like between 26 and 34 degrees Celsius, so it does say use the data. How much do they decrease by? So let's look at this. So it's going to be something times 10 to the fifth. So 845 minus 230, looking at 515. No, what am I? 8 minus, there we go, 615. Huh. So it's just a difference. So 615 times 10 to the fifth. Okay, the growth of that algal species is affected by other factors, including salinity. Outline a lab-based experiment to investigate the effect of salinity on the growth of that organism. Your answer should include a reference to the control variables. So if we want to just maintain or be changing the salinity and nothing else, then things we're going to want to control are going to be temp. And what were they grown in? Did it say test tubes? Okay. Temperature, the nutrient that they put in, because they did the magnesium, they also did the phosphorus. We want to maintain the amount of water in maybe the same type or species of algae. Okay, and the collection, so how will you collect your quantitative results? Guess what? You're going to literally duplicate exactly what they did. Exactly what they did. So you're going to say there, okay, we're going to get, I'll use, what color should I use? Red. Okay. You're going to duplicate it. Just duplicate, duplicate. So they used cultures of that species. You're going to use cultures of that species. And you're going to grow them in test tubes also. And you're going to have a solution inside that test tube of mineral salts. And then you're going to put a starting sample. So at the beginning, you're going to make sure they all have the same amount. Each test tube has 5 times 10 to the 4 cells of that organism. So we'll also include here type of algae and then amount in your sample, because you want to know like how much did it grow. Those are controls. All of the cells were illuminated with fluorescent lights. You're going to do the same thing. So the amount of light they get will be controlled. And we're not going to incubate them. We're going to say all the cells were illuminated with fluorescent lights and given, and within each solution, oh shoot, hold on. Watch this mineral salts. We're testing salinity, so we don't want to include those salts. Not at all. We're not putting mineral salts in there. That's what we're changing. That is what's going to be different. Instead of them, we're going to, like this, the number of cells you're going to use, that's, we're going to count them under the same method that they did. You're going to count the cells using a counting chamber and a microscope. You're going to maintain a standard temperature. So this is going to say, instead, this is going to say salinity. And we measure salinity in parts per thousand. So if you're looking at, like we know average salinity, $35, then maybe this one's 37. Here we'll go 39, 33, 31. And then over here, the same exact thing that they have, the number of cells, right? And then they're all going to be times 10 to the 10 to the 4th, 10 to the 5th, whatever it was going to be. You don't have to assume the results here, but you're going to literally just duplicate it. You duplicate the same thing that they outlined, except like we just saw, pay attention to your control variables. And what you're going to change, the factor you're going to change is either what they give you, or they might say another like abiotic factor or factor that could affect them, and you're just going to try and use that as your manipulative variable. So you'll do the same thing. You're going to see the effect of salinity on the growth of Isochrysis. The experiment, you're going to have cultures of them grown in test tubes. And then the beginning of the experiment, each test tube will contain 5 times 10 to the 4 cells of that organism. All test tubes will be illuminated and given the same volume of water with varying amounts of salinity. The same volume of water, you want the same amount of water, but you are trying to change, you're going to change on purpose the amount of salinity. And guess what? Then after 10 days, same thing, a time factor, you're going to go count those using a counting chamber and a microscope, identical. Results are recorded. And then a safety thing. Yeah. So yeah, I would say like safety. So watch that electrical cord next to the water that you're putting in the test tubes. All right. I just in case you guys see something like that, you got to change a variable tomorrow. Don't be intimidated by it. It's literally just duplicate it. Have a great night.